Hi, welcome to our channel of IGNOU audiobooks, Indira Gandhi National Open University, School of Sciences, SOS, Bachelor's Degree Programs. Bachelor of Science, BSCG. Discipline Specific Elective, DSE. Botany. BBYET 141 Cell and Molecular Biology. Block 1 Techniques in Cell Biology. Unit-1 Microscopy. 1.1 Introduction. Cell biology is the branch of biology which deals with the study of cell structure, their architectural details, major cellular as well as subcellular processes, and the interactive processes between the cells. Various techniques have been used to study the structural details and functioning of the cell. Imaging techniques such as microscopy have proven very useful in studying various aspects related to the architecture of cells, tissues, and various organelles present in them. The minute structural details of the cell, cellular processes, and other characteristics can be easily studied with the help of microscopy techniques. The information procured via these techniques provides a better understanding of the various cellular processes taking place in a living being. Our knowledge about cellular structures and their functions has improved manifold by using microscopy and biochemical techniques. In this unit, you will study about the various aspects of the microscopy techniques and how microscopy helped to understand the structure of cell. An update about various types of microscopy will also be provided to you. Objectives. After studying this unit, you would be able to describe the structure, principle, and functioning of a microscope in detail. Enlist various types of microscopy techniques. Provide details about electron microscopy techniques. Enlist various types of electron microscopy. Describe the preparation of tissue for electron microscopy and describe the applications of microscopy techniques. 1.2 Imaging Techniques, Microscopy. Various techniques are used to study the structural details of the cells and the processes taking place in them. Microscopy is the most common technique used to study the structural details of cells, tissues, and organelles present in plants and animals. A microscope is an instrument used to view small things that cannot be seen with the unaided eyes. The word microscope is derived from the word micro, that means small, and scope, that means to look at. A microscope magnifies objects many-fold so that specimens can be seen easily. The microscopy technique proves useful in visualization of cells and microorganisms as the images appear much larger after magnification. The most commonly used microscope is the optical or light microscope. In this microscope, the glass lenses are used to magnify the image. History of microscope. The concept of magnification came into existence long back in 1267 AD. An English philosopher R. Bacon invented eyeglasses in 1235 AD. In his book, Perspectiva, he mentioned about the small size of dust and sand particles. In 1538, an Italian physician Girolamo Fracastoro observed that an object appears much larger when seen with two spectacle glasses, one superimposed on the other. He mentioned these facts in his book, Homocentrica. The first simple microscope was invented by two Dutch scientists, Zacharias Janssen and his father, Hans, in 1590. They were the first to combine two concave lenses within a tube. They noted that nearby objects appeared closer and larger. They developed a microscope made up of wooden cardboard with an eyepiece and an objective lens. Antony van Leeuwenhoek, a Dutch microbiologist, made first simple microscope in the year 1668. He was able to see the presence of tiny living organisms present in pond water. The organisms appeared like dots. His microscope was made up of a double convex glass lens that was held between two silver plates. Fig. 1.1. Leeuwenhoek was the one who made microbiology popular throughout the world. Fig. 1.1. An image of microscope used by Leeuwenhoek. Single-lens microscopes were first used by English natural philosopher Robert Hooke in 1665. Fig. 1.2. Look at the screen. Fig. 1.2. A. The microscopes used in earlier times by Robert Hooke. B. Image of cover page of Micrographia. Robert Hooke provided information about various microscopic objects, such as fleas, lice, and nettles, in his book Micrographia. He also coined the term cell for small, microscopic structures each living being is composed of. Hence, invention of microscope resulted in the discovery of cell and ultimately the formulation of cell theory. Dutch optician Francois Beelsnijder developed an achromatic, non-color distorting, microscope objective in 1791. In 1827, Scottish botanist Robert Brown demonstrated the presence of nucleus. He coined the term nucleus in 1831. English scientist Joseph Jackson Lister designed microscope lenses free from achromatic aberrations. In 1830, two single-lens microscopes were commonly used in earlier times. Fig. 1.3. In 1868, German physicist Ernst Abbe invented an apochromatic system of lenses. He published lens theory in 1873. A British microscopist Julius Reinberg used color filters in his microscope. The effect came to be known as Reinberg illumination in the 19th century. The largest optical microscope was Birch reflector made in 1947. The weight of this instrument was about 200 kilograms, 440 pounds. The smallest microscope was made by British optician Horace Edward Dall in 1950 that weighed no more than 24 grams, 0.8 ounce. British Dr. John MacArthur designed commercially available small microscope in 1958. Fig. 1.3. Images of two single-lens microscopes used in older times. 1.2.1 Principles of microscopy. A microscope is an instrument which contains two lenses, a light source, and mechanical adjustable parts. We need to be familiar with some terms such as magnification, resolution, numerical aperture, illumination, and focusing before studying in detail about the instrument. Magnification and resolution affect the functioning of microscope to a great extent. The working of a microscope is based upon the basic principle of magnification. Magnification gives a measure of how much bigger an object appears under the microscope. In other words, it is the number of times the object is enlarged when observed under the microscope. You should know that light microscope generally magnifies the object about 400 times the actual size. Magnification of the object depends upon the ocular lens and the objective lens. Resolution. A lens possesses the ability to show two adjacent objects as distinct units. the smallest distance by which two points can be separated and distinguished as individual objects is called resolution. The study of the smallest detail of the object depends on the resolution. Resolution is expressed in micrometers, m, which is 1/1,000,000th of a meter. Factors such as wavelength of light used and the numerical aperture regulate the resolving power of a lens. The diameter of the objective lens in relation to its focal length is known as numerical aperture. The resolution of the microscope is determined by the numerical aperture, NA, of the objective lens. Numerical aperture is also defined as the sine of half the angle of the cone of light taken by the objective multiplied by the refractive index, Ri, of the medium in which the object is immersed. Numerical aperture increases as Ri increases. The values of NA of microscope having low magnification. Objectives range from 0.1 to 0.95 in case of dry objectives and 1.4 in case of all immersion objectives. Magnification. Enlargement. It can be calculated by multiplying the magnification of the objective used. Example 10x eyepiece, 40x objective is equal to 400x total magnification. Numerical aperture, NA is equal to n sin alpha, where n is equal to refractive index of the medium. Alpha is equal to half of the cone angle of light emitted from the condenser. Abbe gave the following formula for calculating resolving power of the microscope. D is equal to lambda to Na, where lambda is the wavelength of light used in visualizing a specimen. Na is the numerical aperture. If using a green light of 514 nanometers and an OI immersion objective with an Na of 1.45, the theoretical limit of resolution will be 177 nanometers. It has been noted that resolving power of the lens is higher if the wavelength is short. Hence, smaller the value of resolving power, higher the resolution and more the clarity. Box 1.1 Resolution. The resolution of an optical system is calculated using the formula R is equal to 0.61 lambda n sin alpha. Where R is the resolving power, it is the minimum distance between two points that can be separated. Lambda is the wavelength of light used to illuminate an object. N is the refractive index of the medium in which the object is placed, and sin alpha is the sine of half the angle between the specimen and the objective lens. The term n sin alpha is often referred to as the numerical aperture. 1.2.2 Light microscopy. You might have used microscopes in the laboratories. These are called as light microscopes or optical microscopes. In this type of microscope, visible light passes through the specimen, the biological sample, and is bent to form a magnified image. Lenses focus light on the specimen, magnifying it, thus producing an image. The magnification varies depending on the types and number of lenses used in the microscope. Optical microscopes are of two types: simple or compound. Times simple light microscope. A microscope having one lens is called a simple microscope. This type of microscope can magnify an image up to 300x. It has low magnification as it uses a single lens. The resolution power of a simple microscope is below 1 m. Compound light microscope. A microscope having more than one lens is called compound microscope. This type of microscope can Magnify images up to 2000x. It has a higher magnification as it uses two sets of lenses, an objective lens and an eyepiece. The lenses are aligned in such a way that they can bend light for getting effective magnification of the image. The resolution of compound microscope is about 0.2 m. The limitations of resolution, magnifying power, found in a simple microscope can be overcome using this microscope. The magnification of the objective lens and eyepiece manage the magnifying power of the compound microscope. Parts of a microscope. A microscope mainly consists of many parts. These include an objective lens, ocular lens, lens tube, stage, and reflector. An object is magnified through the objective lens. When the target is focused, a magnified image can be observed through the ocular lens. Objective. The objective lens gathers light from the specimen, magnifies the image of the specimen, and projects this magnified image. Objective lenses show magnifications in the range of 2 to 100x. Objective is designed to minimize aberrations. Objective can be of two types, dry or immersion. An objective which works with the air between the specimen and the objective lens is called as dry. In contrast, an objective that requires liquid and is usually transparent, all which occupies the space between the object and the front element, is called as an immersion objective. In the first type of objective, refractive index, RL, of foil matches that of the glass. The focal length regulates the optics of the objective. The focal length is inversely proportional to the magnification. This means that focal length of an objective decreases with the increase in magnifying power. In some objectives, special glasses are used. These are called apochromats and were first produced commercially by Abbé in the 1870s. Aberration is noted in high magnification objectives, magnifications greater than 25x. Aberration influences the sharpness or quality of the image. The aberrations of a lens increase as the relative aperture of the lens is increased. Eyepiece, ocular lens. Eyepiece is the lens which is used to observe the image. These lenses are usually of 10x or 15x magnification. The eyepiece helps in forming enlarged virtual image that can be seen by the observer. Body tube. A hollow tube through which light travels from the objective to the ocular lens is called as body tube, Fig. 1.5. It connects eyepiece to the objective. It contains a prism that bends the light rays so they can enter the tube. The upper end of body tube possesses ocular or eyepiece lens, while the lower portion consists of a movable nosepiece containing the objective lenses. Look at the screen. Arm. It is a structure that connects head of the microscope to the base. Base. The bottom of the microscope is called base. Stage. It is a flat platform having clips that supports the slides, Fig. 1.4. In case of mechanical stage, the slide kept in position with the help of two knobs. One knob moves the slide left and right, the other moves it forward and backward. Nosepiece. The circular structure that screws the objective lenses is called nosepiece. Objective lenses of varying magnification and numerical aperture are mounted on the rotating nosepiece. Condenser lens. The condenser is mounted below the stage where the specimen is placed and focuses beam of light onto the specimen. When it provides uniform illumination in the region of the object under observation, it is referred to as critical illumination. Alternatively, when the image is focused on the condenser, followed by objective of the microscope, It is referred to as solar illumination. It is useful in getting sharp images of magnification 400x and above. For a microscope with a power of 1000x or above, condenser lens with an NA of 1.25 or greater is needed. Such microscopes are generally equipped with an Abbe condenser, diaphragm or iris. The diaphragm or iris regulates the amount of light entering the condenser, i.e., helps in adjusting the intensity of light. It is located under the stage or between the light source and condenser. Illuminator. A steady light source is used for illumination in the microscope. Mirrors were used as a source of light in early microscopes. Mirror reflected light up from an external light source located at the bottom of the stage. Generally, a tungsten lamp is used as a light source. Effective illumination is required for getting good magnification and resolving power. The amount of light entering the microscope needs to be regulated as excessive illumination may obscure the specimen. Box 1.2 Refractive Index. The resolving power of microscope depends on the refractive index, Ri. Refractive index is the capacity power of light to bend while passing through air from the glass slide to the objective lens. The refractive index of air is lower than that of glass. When the light rays pass from the glass into the air, they bend or refract so that they do not pass into the objective lens. Loss of refracted light can be compensated by mineral oil, which has the same refractive index as glass. The loss of light reduces the numerical aperture and resolving power of the objective lens. The decrease in light refraction produces more light rays which enter directly into the objective lens, producing a clear image with high resolution. Electronic detectors such as a complementary metal oxide semiconductor CMOS or charge-coupled device CCD chip is used to capture the magnified image in modern-day microscopes. The digital signal obtained is transmitted to a computer and translated into an image on the monitor. The pictures as well as moving video sequences are captured using software. The specimen is mounted on a glass slide and immersed in a material which has the same Ri as that of the slide. The tissues are cut into thin sections. After treatment with fixatives, the preparations of this type preserve the natural structural appearance of the cells. The acids and aldehydes such as formaldehyde, acetic acid, and picric acid are widely used as fixatives. The thin sections of the tissues are cut after getting them embedded in paraffin, wax or quick frozen. The sections are cut using an instrument called microtome. The thin sections are spread on the slides and stained. The sample preparation is such that contrasts between the subcellular structures as dark or light or colored areas are produced. Contrast is produced by applying specific stains which give color to different structures or by bringing alteration in light transmitting properties of cell components. Box 1.3 Abbe theory of image formation in a microscope. The modern theory of image formation in the microscope was given by German physicist Ernst Abbe in 1873. According to this theory, convergent light obtained from a condenser helps in visualization of objects or specimens in the focal plane of the microscope. A collection of plane waves propagating in a specified set of directions is called as convergent light. The superimposition of waves produces incident Illumination. The diffracted waves are collected by the objective and directed to the focal plane. Interference between the diffracted waves produces an image of the object. Abbe showed that more is the number of diffracted waves reaching the objective, finer is the details of the image. The objective's ability to collect diffracted light was considered as the NA. Greater is the magnification of the objective, greater is NA of the objective. Specialized optical microscope. The modifications and a range of special adaptations have been made in basic optical microscope. In some specialized microscopes, the illuminator and lens systems are incorporated into a single unit. Inverted microscope. The light source and condenser situated in the upper part of the microscope. Direct light down to the stage. The eyepiece in this instrument is angled upward while the objective is set with its front element. The specimens present in the liquid medium can be studied with the help of this microscope. The cell cultures have been studied using this microscope. Hence, the microscope has proved useful in studies related to biology and medical research. Stereoscopic microscopes, dissecting microscope. The microscope possesses two objectives and eyepieces. They provide separate optical paths and different viewing angles to eyes. This arrangement produces a three-dimensional visualization of the sample. Each eye can independently image the object. The magnifying power ranges between 5 and 250x. This microscope is generally used for viewing the dissected subjects and microsurgical procedures. They are also used in electronics manufacturing units to monitor the bonding of leads to integrated circuits. Metallographic microscope. In this type of microscope, vertical source of light, illumination, is used. The light source is generally present below the eyepiece in the tube. Light is focused through the objective onto the specimen. The reflected light or scattered light gets back to the objective, which is imaged back at the eyepiece. This type of microscope is used to identify defects in metal surfaces, determine crystal grain boundaries in metal alloys, and study rocks and minerals. They also have applications in forensic science and diagnostics. Reflecting microscope. Microscopes of this type possess reflecting objectives. An objective usually consists of two components, a relatively large primary concave mirror and a smaller secondary convex mirror which is located between the primary mirror and the object. The objective lens of this type does not produce chromatic aberration and needs no correction. The objects can be observed over a wide range of visible light and also in the ultraviolet or infrared regions. Bright field microscope. Two lens systems are used for magnifying specimens. This includes ocular lens located in the eyepiece and objective lens located in the nosepiece. The beam of light obtained from tungsten lamp helps in illumination of the specimen. The light is focused on substage lens called a condenser. This microscope provides a high-resolution image. These microscopes are used for observing non-viable, stained samples. We cannot observe living cells because of the absence of contrast between the specimen and the surrounding medium. Dark field microscope. In this type of microscope, the condenser is modified which directs light obliquely on the specimen. This results in deflection or scattering of light. hence making object appear bright against a dark background. This type of microscope is suitable for observing living specimens. Interference microscope. Two beams of light are used in this microscope. One beam of light passes through the specimen while the other bypasses it. The combination of two beams results in interference between them, thus revealing the structure of the specimen. The difference between interfering beams of light is used to create the image. This type of microscopy was developed by British microscopist Francis Smith and French physicist Maurice Franckon in 1947. Quartz lenses were used by them to produce two beams of light that were perpendicularly polarized. 1.2.3 Phase contrast microscopy. Phase contrast microscope was invented by Dutch physicist Frits Zernike in 1934. He was awarded Nobel Prize in Physics in 1953. In this type of microscope, a cone of light is passed through the specimen, which gets diffracted. The light rays pass obliquely through the specimen, causing refracted waves to deviate away from the unrefracted rays. Fig. 1.6. The unrefracted and refracted rays are focused back into the image plane. Two or more light waves combine to produce interference. This happens because wave equal to the sum of the two combining waves is produced. The direct and undeflected light creates a difference in phase producing contrast. The phase difference of about 1/4 wavelength between two waves produces only minimal interference. Fig. 1.6 Graphic explanation of principle of the phase contrast microscope. In this microscope, delay in refracted rays by about 1/4 wavelength than normal light microscope has been noted. A comparison of the diagrams on the right illustrates that extra delay enhances the interference obtained when the unrefracted and refracted rays are focused back together, U plus R. The problem of staining and alterations in brightness can be overcome by this microscopy. The technique proves useful for observing colorless specimens. The transparent structure such as nuclei transmits a light when present in the mounting medium that surrounds them. The propagation of light through object produces a change in the optical path. The shift in the phase of light passing through the structure and that passing around the structure forms the visible image. 1.2.4 Fluorescence microscopy. Fluorescence microscope is used to see tissues that fluoresce, absorb one wavelength of light and emit another. Light of one wavelength excites the fluorescent molecules while the light of other wavelength collects the emitted light. Some cellular components fluoresce naturally and appear in various colors, while non-fluorescing structures fluoresce by staining with fluorescent dyes, fluorochromes. Fluorescence dyes are used in preparation of antibodies that bind to specific cellular proteins. The antibodies are first tagged with fluorescein, a fluorescent dye, and then fluorescein labeled antibody is applied to cells. 1.2.5 Confocal microscopy. Confocal microscopy is an advanced microscopy technique which uses a pinhole to eliminate out of focus light. The discrete optical sections can be collected after eliminating out of focus light above and below the plane. High power lasers illuminate the sample. They help in collecting light from the desired focal plane. The beam of laser light gets scanned across the sample, and light only from the desired focal plane can enter the detector. This Microscope uses fluorescence optics. Fluorophores, i.e., a fluorescent chemical compound that can re-emit light upon excitation, is used in the sample. Fluorophores contain aromatic groups, planar or cyclic molecules with several pi bonds. When the laser light gets focused on a defined spot at a specific depth located within the sample, fluorescent light gets emitted exactly at that point. A pinhole cuts off out of focus signals from the optical path, thus allowing the fluorescence signals from the illuminated spot to enter the light detector and form image at different depths in the same sample. The technique is suitable for both live as well for fixed cells and tissues. Fig. 1.8. An image showing the working of a confocal microscope. 1.2.6 Polarization microscopy. In polarization microscope, light source is equipped with a polarizing filter. The linearly polarized light, whose wave travels in a single plane, passes through the object. It either remains unaffected or splits into two beams with different polarizations. The polarized light improves the contrast. Polarizer is a device that transmits polarized light through the specimen. Analyzer is a device placed above the objective. An analyzer fitted to the eyepiece blocks polarization of the light. The analyzer is rotated to get maximum contrast in the image and a difference in refractive index between planes is determined. Polarization in the beam of light is removed by using objectives with low numerical aperture. The differences in refractive index provide useful information about the molecular substructure of cell organelles. This microscopy is useful for visualizing birefringent structures which differ in refractive index in different planes. The crystalline substances, geologic samples and fibrous materials such as the mitotic spindle and muscle fibers can be easily studied using this microscope. 1.3 Electron microscopy, M. Electron microscopy, M, is a technique that helps in getting high-resolution images of specimens, biological and non-biological. A beam of electrons is used as the source of illumination. The electrostatic and electromagnetic lenses are used to control the beam of electron and focus it to form an image. Basic components of an electron microscope. 1. The vacuum system creates strong vacuum in the entire column i.e. along the path of electron beam. This is required for making the electrons travel as they do not travel easily in air. 2. Electron gun is used for emission of electron beam. Electron gun consists of A. Cathode, which is a filament made of tungsten that emits electrons at 50-100 kV. B. Anode that produces a powerful electric field. The hole in anode provides an electron beam which makes the electrons travel at a high speed, several 100,000 kilometers per second. The voltage applied is the range of 3 to 5 kilovolts. Voltage is required to draw electrons from the source. 3. Electromagnetic lenses are arranged together. They help in illumination, focus, and magnification. The condenser lens controls the electron beam. Objective lens, intermediate lens, and projector lens work together to produce a final image. Fig. 1.9. 4. The photographic system is used to record image as an electron micrograph. 5. A cooling system attached to the column prevents heating up of system, since high voltage is used for emission of electrons. This microscopy is widely used in biomedical research and study of detailed Structure of tissues, cells, organelles, and macromolecular complexes. Look at the screen. Fig. 1.9 Schematic diagrams of light and electron microscopes showing difference in the principle of working. Electron microscopy is of two types: transmission electron microscopy (TEM) and scanning electron microscopy (SEM). In TEM, the electrons are transmitted through an object, focused by the lenses to form the image, while in SEM, electrons are reflected by the object to form an image. 1.3.1 Transmission electron microscopy (TEM). The transmission electron microscope possesses high resolution. The concept of electron microscopy was given by German physicist Ernst Ruska and the electrical engineer Max Noll in 1931. The first functional electron microscope was made by E.F. Burton and his students C. Hall, J. Hillier and A. Priebus at the University of Toronto in 1938. Later in 1939, the first commercial TEM was developed by Siemens. In this type of microscope, a beam of highly focused electrons is directed towards a thin section of the specimen, less than 200 nanometers. The highly energetic incident electrons interact with the atoms in the sample and produce characteristic radiation and particles which form image. A high-voltage electron beam is emitted by electron gun, which is made up of a tungsten filament. The cathode is used as the electron source, while acceleration of electron beam is produced by anode. Fig. 1.10 The electron beam is focused by electrostatic and electromagnetic lenses. The transmission of electron beam through the specimen results in magnification of the structure of the specimen through objective lens. The transmitted electrons hit a fluorescent screen at the bottom of the microscope and give rise to a shadow image of the specimen. The image is projected onto a fluorescent viewing screen coated with a phosphorescent or scintillator material. The photographic film or plate is exposed directly to the electron beam or a fiber optic light guide to the sensor of a CCD camera. The image detected by the CCD may be visualized on a monitor or computer. The sample specimen needs to be processed before viewing under an electron microscope. The material sample is prepared by different ways. This is done either by cutting very thin sections of the specimen from a piece of tissue after fixing it in resin, or freezing it, or isolating a specimen and studying it after negative staining. Fig. 1.10 A. Photograph of a TEM. B. Schematic representation of TEM showing different electromagnetic lenses. Sample preparation. As the TEM works under conditions of vacuum, water present in biological materials needs to be removed. The tissue is preserved using different fixatives to avoid tissue disruption caused by this loss of water. These fixatives stabilize the macromolecular structure of specimen by cross-linking of proteins with aldehydes such as formaldehyde and glutaraldehyde, and lipids with osmium tetroxide. The tissue is dehydrated using alcohol or acetone and embedded before sectioning. The tissue is passed through a transition solvent such as propylene oxide and then infiltrated with an epoxy resin such as araldite, epon or dercupin. The resin gets polymerized, hardened, and the hardened sample is cut into very thin sections using a diamond or glass knife present in the instrument called ultramicrotome. The thin sections are picked up using a boat filled with water. made present around the glass knife. The cut sections float on the surface of water, which are picked up and placed on the surface of copper grid, Fig. 1.11. The staining is done with heavy metals such as lead, uranium, or tungsten. The scattering of electrons produces contrast between different structures. The specimens can be stained en bloc before embedding or later after sectioning. In en bloc staining, a fixed biological specimen, block tissue, is immersed in an aqueous solution of uranyl acetate for several minutes and are followed by aqueous lead citrate and or lead aspartate. This process enhances the image contrast of membrane structures or fibrous structures in cells. A microtome is a tool used to cut extremely thin sections of specimen sample. The word microtome is derived from Greek micros, meaning small, and temnin, meaning to cut. It is also known as an ultratome or ultramicrotome. The instrument is used in the preparation of ultra-thin sections 50, 100A that can be observed under TEM. Glass and diamond. Knives present in the instrument help in cutting very thin sections. TEM shows enhanced resolution, but the electron beam used is very weak and pass an appreciable distance through air, therefore high vacuum is required. Inside the internal chamber of electron microscope, the photographs of cells taken using an electron microscope are called electron micrographs. You can see that the picture below shows the minute structural details of the cellular entities, Fig. 1.13. Fig. 1.13. Electron micrograph showing to plant cells, X45, 000. CW, cell wall, ER endoplasmic, M mitochondria, PM plasma membrane, PP proplastids, tetonoplast. 1.3. To scanning electron microscopy, SEM. In scanning electron microscope, electron beam moves over the specimen. The molecules get excited and move to high energy level and emit secondary electrons. These electrons are used to form image of the specimen. Image is produced when electron beam is focused into an intense spot on the surface of specimen. The spot is moved back and forth across the specimen by charged plates called beam deflectors. These deflectors are located between the condenser lens and the specimen. The signals sent by the deflector attract or repel the beam. Secondary electrons are captured by a detector. The scintillator emits photons of light on excitation when electrons are incident upon it, Fig. 1.14. The deflected and emitted electrons are detected by a photomultiplier tube. This tube forms a 3-D image of surface features of the specimen. The electronic signal generated is transferred to the video screen. Fig. 1.14. Scanning electron microscope. Generally, the image resolution of SEM is less than that of TEM. The SEM image depends upon the surface processes. SEM can produce images of bulk samples up to many centimeters in size. It produces images of three-dimensional shape of the sample, Fig. 1.15. This microscope has proved very useful in studying the minute structural details of the cell surface, Fig. 1.16. The increasing popularity of SEM among cell biologists is because of its remarkable ability to provide details about surface topography, show 3D structure and improved resolution, 30 to 100A. Fig. 1.15. Schematic diagram of a SEM. Sample preparation. The sample preparation for SEM involves various steps. 1. The material is fixed by immersing specimen in 2.8% glycerol. Prepared in 0.1 m Heps buffer, pH 7.2, having 0.02% triton x 100, for several hours at room temperature or overnight at 4 degrees C. 2. The fixed specimen is washed thrice, for 5 to 10 min, in 0.1 m Heps buffer, pH 7.2. 3. The specimen is dehydrated by treating it with 25% ethanol for 10 minutes, 50% ethanol for 10 minutes, 70% ethanol for 10 minutes, 85% ethanol for 10 minutes, 95% ethanol for 10 minutes, and 100% ethanol for 10 minutes. 4. The automated process of critical point drying is carried for 40 minutes. 5. The sample is mounted on metal stub with double-sided carbon tape. 6. The sample is coated by applying a thin layer of metals, gold and palladium, using an automated sputter coater. 1.3.3 Scanning Transmission Electron Microscopy. STEM. Scanning Transmission Electron Microscope combines elements of both TEM and SEM. STEM is a sophisticated technique and requires high vacuum conditions. It is electronically more complex than TEM or a SEM, Fig. 1.17a. STEM is equipped with scanning coils, detectors and necessary circuitry. The image is formed by the secondary electrons since the primary electrons transmit light across the specimen. The electron beam is focused to a fine spot, size 0.05 to 0.2 nm, which is then scanned over the sample. The magnetic lenses focus beam of light that seeps over the surface of the specimen, Fig. 1.17b. The sample is illuminated at each point with the beam parallel to the optical axis. The technique of STEM helps in distinguishing specific characteristics of the electron transmitted by the specimen, thus providing unique information about the specimen. Fig. 1.17a. Scanning Transmission Electron Microscope, STEM, B. Schematic diagram of STEM. 1.3.4 Environmental Scanning Electron Microscopy, ESEM. Environmental Scanning Electron Microscope, ESEM, is a specialized SEM in which uncoated specimens are analyzed. The first commercial ESEM was produced by the Electroscan Corporation in USA in 1988. The instrument produces high-quality and resolution images in wet samples and those contained in low vacuum or gas, Fig. 1.18. The samples present even in low-pressure gaseous and high humidity, up to 100%, conditions could be observed clearly. The technique enables the imaging of biological samples unstable in high vacuum conditions, conventional electron microscopes. This is because this microscope possesses a secondary electron detector which is capable of operating in the presence of water vapor. The pressure-limiting apertures separate the vacuum region from the sample chamber. The specimen is placed in a relatively high-pressure chamber and the electron optical column is differentially pumped to keep vacuum adequately low at the site where the electron beam is exposed. 1.3.5 Comparative account of different types of microscopes. Each microscope shows variations in its structure, technical details and produces images of characteristic type. The details about various microscopic techniques have been provided to you in the sections above. A summary of comparative account of the characteristics of different microscopes has also been given to you for more clarity. Table 1.1 and Fig. 1.20. Table 1.1 Comparative account of different types of microscopes. Look at the screen for table content. Fig. 1.20 Image micrographs of an algal cell as seen under different microscopes. A. Compound. B. Confocal. C. SEM. D. TEM. 1.4 Techniques for preparing tissues. For electron microscopy. Technique of electron microscopy helps in characterizing the microstructural features of a sample ranging from 100 nanometers to 100 m. Various methods pertaining to processing preparation of samples have been developed over the years. 1.4.1 Negative staining. Negative staining method is used for examining very small objects. It is generally used in TEM. The shape and surface characteristics of intact organelles or viruses can be examined using this technique. In the method, thin sections are not cut but particles are suspended in a small drop of liquid, applied to copper grid and allowed to dry in air. The dried surface of sample is treated with stains such as phosphotungstic acid or uranyl acetate, Fig. 1.21. The specimen is visualized against the stained dark background. 1.4.2 Freeze fracture. The technique involves physical breaking, fracturing, of a frozen biological sample along the planes of natural weakness that run through each cell. These planes generally occur between the two layers of lipid molecules which forms part of membrane and around various organelles of the cell. The main steps involve in formation of a freeze fracture replica are I. Rapid freezing, 2. Fracturing, 3. Formation of replica, and 4. Replica cleaning. The pretreatment carried out before freezing comprises fixation of material in glycerol followed by cryoprotection with glycerol. Cryoprotection is done to reduce the formation of ice crystals during freezing. This kind of sample preparation provides planar views of the internal organization of membranes. Images provided by these samples have proved useful understanding the functional morphology of the cell. This technique has been used to study membranes and reveal the pattern of integral membrane proteins. General outline of technique. I. Cells are frozen in liquid freon, minus 130 degrees C. This results in instant immobilization of cell components. 2. The block of frozen cells is fractured in an evacuated chamber. The fracture is irregular and occurs along lines of weakness like the plasma membrane or surfaces of organelles. 3. A replica of the fractured specimen is made by shadowing with platinum. Fig. 1.22. 4. A carbon replica is produced when a thin layer of carbon is evaporated vertically onto the surface. Replica is left behind after digestion of organic material by acid. 6. The replica is studied under M. Fig. 1.22. Diagrammatic representation of various steps of freeze fracture. Technique I6. 1.4.3 Freeze etching. The freeze etching technique of sample preparation is related to freeze fracture. One more step is added to freeze fracture procedure. In this method, the specimens are rapidly frozen in liquid freon under vacuum and struck with a sharp knife edge as in freeze fracture method. Biological samples fracture along lines of natural weakness at low temperature. The tissue is subjected to vacuum which results in sublimation of water from the fractured surfaces. This removal of water produces an etching effect. This etching will cause small areas of the cell surface around the periphery of the fracture face to stand out against the background. A replica of the specimen is made by heavy metals such as platinum, followed by backing it with a carbon film. After dissolving the tissue in strong acid, the remaining metal replica can be viewed with the electron microscope. Such preparations provide a unique picture of cells. This procedure is useful because it avoids exposure to fixatives, embedding agents, and stains which can deform ultrastructure of cell, Fig. 1.23. This type of specimen treatment causes minimal tissue distortion and permits immediate arrest of cell function. 1.4.4 Shadow casting. Shadow casting is a technique which helps in studying surface texture of the specimen material. The method involves in vacuum deposition of a metallic film on dried specimens. A three-dimensional electron beam image is produced using this technique. This process involves deposition of an electron dense substance upon the specimen at oblique angle so that only a part of the specimen is coated. Fig. 1.24. The deposition of metal on one side creates a shadow, resulting in three-dimensional appearance. Fig. 1.24. A. Technique of shadow casting. B. Vacuum evaporator. Look at the screen. The thin metal film is formed on the specimen by condensation after vaporization. The metals with the high vaporization temperature condense quickly after vaporization. The isolated particles can be visualized by placing them in an evacuated chamber and spraying heavy metal across their surfaces. The particle size of a film of evaporated gold will be larger than that of platinum or palladium. The grain size of evaporated tungsten is exceedingly fine, but its deposition time is long. The concurrent evaporation of two or more elements results in smaller aggregate size by increasing the distance over which any atom diffuses within a crystal lattice. 1.4 Summary. Times Cell biology is the branch of biology that provides information about cell structure and function. A combination of microscopic and biochemical techniques has enhanced our knowledge and understanding of cell structure and function. Times the discovery of microscope has proved as a milestone in the study of cell biology as minute structural details and major cellular mechanism could be revealed by this technique. Times initially, light microscope proved very useful in studying the structural details of cells, though lower resolution, 200 nanometers, restricted the study of detailed cell architecture. Times various other versions such as phase contrast, confocal and fluorescence microscopes developed over the time provided a medium of studying structural details of the cell sample specimen present in any medium or form. Times the change of source of illumination from light to electrons significantly improved the resolving power of the microscope. The invention of the transmission electron microscope improved our understanding of cell biology, but the sample to be viewed required preparation before use. Times various advancements such as transmission electron microscope (TEM) and scanning electron microscope (SEM) proved very useful as they showed remarkable ability to study surface topography and 3D structure, along with improved resolution 30 to 100 A. The electrons are transmitted through an object and then focused by the lenses to form the image in TEM, while the electrons are reflected by the object in A. scan pattern which forms the image in SEM. Times Scanning Transmission Electron Microscope (STEM) contains elements of both TEM and SEM. It provides unique information about the specimen. STEM requires high vacuum condition and electronically more complex than a TEM or a SEM. This microscope is equipped with additional scanning coils, detectors, and necessary circuitry. Times a specialized SEM, Environmental Scanning Electron Microscope (ESEM), performs analysis using uncoated specimens. The samples are observed in low-pressure gaseous and high relative humidity, up to 100% environments. It produces quality images and gives good resolution, even for wet samples or those contained in low vacuum or gas. Times diverse set of procedures for specimen preparation, such as thin sectioning, negative staining, positive staining, shadow casting, hole mounting, and freeze fracture has been developed so that minute architectural details, three-dimensional view of the cell surface can be captured, and details of the cellular processes can also be revealed. Thank you. Subscribe to our channel for more updates, and we will see you with the next chapter.